それではお時間になりましたので再開させていただきます。これよりパネルディスカッション1、COVID-19, Our Experiences and Lessons Learned in Preparation for Future Pandemics。COVID-19、我々は何を経験したか、何を考えるべきかを開始させていただきます。モデレーターは AMED スカーダ最新的研究開発戦略センター、浜口道成センター長です。パネリストはボストン大学国立新興感染症研究所所長ナンシー・サリバン様、国立感染症研究所副所長又野哲郎様、感染症流行対策イノベーション連合 CEO リチャード・ハチット様にご登壇いただきます。それでは浜口センター長、パネリストの皆様、どうぞよろしくお願いいたします。
Thank you very much for introduction. I'm Hamaguchi, the Director General of the Advanced Research and Development Strategy Center, so-called SCADA. Thank you for all the members joining this panel discussion, and I'm deeply appreciate foreign panelists, despite the very busy schedule, joined us today. As you may remember, let me start. Slide, please. As you may remember, COVID-19 emerged at the end of 2019, 5 years ago. That has spread worldwide without vaccine and drugs and causing significant damage to the world. This slide shows the latest number of deaths due to COVID-19 published by WHO. Over 7 million people have passed away by the outbreak. It's really pity. Based on our painful experience, SCADA was established 3 years ago. As shown here, SCADA aimed to create vaccines smoothly and effectively to save people and prepare for the next pandemic. To ensure that we shouldn't forget the lesson from COVID-19 and to learn more for next pandemic, we plan today's session. Let me introduce the panelists first of all. Dr. Nancy Sullivan just lied to me. She is a professor at Boston University in the United States and also serves as a director of the National Emerging Infectious Diseases Laboratory, NIDL, established at the university. As an academic researcher, she has led the development of Ebola virus vaccine, very, very dangerous, lethal virus. Moreover, even before the COVID-19 pandemic, she has been advocating the importance of pandemic preparedness based on her field experience. I believe she will share her experiences, including the challenge of vaccine development, field experience, and more importantly, approaches to infectious disease based on virus classification, function, and structure. This is, I would say, prepare the preparedness based on the structure and function of virus. This is the first talk. Second panelist is Dr. Tetsuo Matano. He is the Deputy Director of the National Institute of Infectious Disease in Japan. He began his research on HIV at the National Institute of Allergy and Infectious Disease in the United States, where he has been working for the development of vaccines. As of coming this April, it has been decided that the National Institute of Infectious Disease, NIID in short, and the National Center for Global Health and Medicine, NCGM in short, will merge into the Japan Institute of Health and Safety, JIS in short, which will be responsible for strengthening pandemic responses. I believe he will discuss that response during COVID-19, the current situation, and the function that JIS will carry out in the future. This is a kind of preparation for the preparedness through the systemic innovation. Because when COVID-19 started, we couldn't make any kind of vaccine smoothly. We think it's mainly based on our science research system. Japan is a kind of silo society. We have everything focused on one issues, so we cannot collaborate well. This weakness made our failure. So his approach is a kind of structural innovation to change our system. Anyway, preparation by systemic innovation. Last panelist is Dr. Richard Hazzard. He's the CEO of CEPI, a vaccine development funding agency based in Europe. As a CEO of CEPI, he has been demonstrating leadership in pandemic preparedness and vaccine development. Since 2021, he has been one of the leaders promoting the 100-day mission started at the UK's G7 summit, which aimed to develop and provide a new vaccine within 100 days of the recognizing a new pandemic threat. I believe he will share with us the current state of global infectious disease response and the tough reality of collaboration across the different values and countries in the time of decoupling. Now, each panelist will give a short presentation, roughly 20 minutes, and rest of the time we will have discussion. So first, we have Professor Dr. Nancy Sullivan from Boston University, please.
Thank you very much, Dr. Hamaguchi, and thank you to AMED for inviting me to this really special event, the 10th anniversary of AMED, which I think we can all appreciate how important AMED and SCARTA are based on the fact that we've all just been through a pretty devastating pandemic. So I'd like to just today take a short time to review what we've been thinking are the important aspects of preparation scientifically that will help us so that we are not experiencing pandemics every 5 years or so. As Richard will tell you, it seems to be quite frequent. And so basic research really is the pillar of that pandemic preparedness. And I'm going to start by telling you just some of the personal experiences with Ebola that led me back in the late 1990s to realize that we need to be preparing ahead of time for these things that are out there and scary, but have the potential to cause great harm. And so in the time between the discovery of Ebola in the mid-1970s and the time that I began studying Ebola, there had been many attempts to develop a vaccine against that virus because I wasn't the only one thinking that it could be quite deadly if we were to have an epidemic. And most of those approaches used what was common at the time, and that is recombinant protein. And the recombinant protein technology really took off in the 90s as a tool for making vaccines. And what was interesting was that for all of those studies using protein, they were able to generate high amounts of neutralizing antibody levels in animal models, but that didn't translate to protection in either mouse models or primate models. And in some cases, in the guinea pig model, those vaccines led to enhanced disease. So that was very puzzling. The next thing that people tried was plasmid DNA vectors, and I'll talk to you a little bit in a moment about the different kinds of vaccines, but those weren't quite as good at developing high antibody levels, but nonetheless, there was no protection. So it made me think, as a scientist just coming into the field, maybe we need to think about the basic biology of this virus to understand how we need to develop vaccines. And the first thing that I thought about was it's a filovirus. It's named for this filamentous structure. It's a very large virus, so it's 14,000 nanometers, which is probably 10 or 20 times the size of an HIV particle. Because it's pleomorphic, it folds on itself. So it's got a high density of glycoprotein, which would be the target of vaccine-induced antibodies. And by that folding on itself, it may be that some of those glycoproteins aren't even accessible to antibodies. So it's a tough problem to start. The other thing that is tough about Ebola is that it's quite a stable virion, and it causes a lethal infection with less than one plaque-forming unit. In the monkey model, it's 100% lethal with a very small amount of virus. And not only that, but it's got a broad tropism. So unlike some viruses that might concentrate in the nasal passages or the lungs, Ebola is tropic for virtually every tissue in the host. So if you wanted antibodies to protect, you'd need a lot of antibody to coat the virus, and you'd need it everywhere. The other thing that is a problem is that the virus is rapidly internalized, so it's only available for antibody binding for a short time. So that got me thinking that maybe antibodies aren't enough for protection against this virus, that we would need a vaccine that could also develop a potent T-cell response. And so I'll take a moment just to go through the different vaccine technologies, because it really is quite important as we think about pandemic preparedness. What we want is a set of vaccines on the shelf that we can choose from depending on what pathogen is emerging. And so, as I mentioned earlier, there are some technologies that have been around for a long time. Two of them are attenuating viruses, just weakening them, usually using some sort of chemical modification, or completely inactivating them. Attenuated viruses tend to generate very broad immune responses. So you get cellular responses in the form of CD8 or CD4 T cells, and also antibody responses. Inactivated viruses, on the other hand, since they're not replicating, are pretty good at generating antibody responses, but not the cellular responses. And then as we move through the years in developing different vaccine platforms, DNA plasmids became quite popular for testing and are really efficacious against some viruses. And DNA tends to generate that cellular response that attenuated viruses do, maybe not quite as high. Antibody responses aren't quite as good. You're getting the picture here that the different vaccines are doing different things to the host immune response. Recombinant proteins generate very potent antibody responses, but almost no T-cell response unless you give them repeatedly. Virus-like particles are an evolution of the recombinant proteins, where you can make a recombinant virus-like particle by combining lots of proteins. Gives a bigger antibody response, a little bit of cellular response, but still not as good as what you might see with an attenuated virus, or the next generation, which is viral vectors. And that is taking the genetic material of the virus that's your target and delivering it with a viral vector. That can be a live vector, it can be replicating, but it's not the same as the virus that's infecting. And then finally, you're all familiar with the mRNA vaccines, which were really saving the world, I'd like to say, without being too dramatic, but it really was the mRNA vaccines that controlled the pandemic. And those are good at developing antibody responses, but not so much on the T cell responses. And the reason I'm focusing on these immune responses is you can get a very fast immune response the way we did with the mRNA vaccines, but they tend not to be durable. So at the end of the day, we want something that does both. And when I was thinking about Ebola virus and the need for T cells, I was looking at two platforms that can tune the immune response toward both antibodies and T cells, and that's DNA and viral vectors, and it was called a heterologous prime boost. So when you give these two things together with some distance in between them, they really generate about the best immune response and balanced that you can imagine. And this led to the development of a vaccine that was later put through clinical trials. And I think what I'd like to emphasize here is that this preclinical work took place over many, many years. And that was followed by an additional number of years for the clinical testing that would allow us to move these into humans when there was an outbreak in 2014. Julie Ledgerwood, a very talented clinician who was at the Vaccine Research Center with me at the time, actually developed these phase 1 trials that enabled us to change the insert of the vaccine, change the vector of the vaccine, leading to a large number of safety studies that helped in the 2014-2016 outbreak. But the point I'm trying to make here is one that Jeremy and others have made, and that is the need for doing all of this basic research so that you're ready when something comes along. And so when I published the first paper about the Ebola vaccine, there was a piece in the Wall Street Journal where the journalist said, well, who cares? This only kills a few hundred people in Africa every decade or so. Why should we care about this virus? And it wasn't until 2014 when the Wall Street Journal had another article, now it's in the middle of an outbreak, where they thought, okay, this is important. And at that time, President Obama came to my laboratory and I was showing him my notebook from 1999 to impress upon him how many years it took to get to the point that these vaccines could be mobilized. And he rightly noted, I guess we don't know we care until we do, and this is why we need basic research and we need to support it. So this is the real importance of AMED and SCARTA and CEPI and WHO and all of the other organizations that are moving us along in these areas. So now that's Ebola. That's one story with a couple of decades that were required to develop a vaccine. Now we look at all of the emerging infections since 2003, and it's hard to even take in this graph. So the red pathogens are diseases that are new to an area, not necessarily new, and the blue are newly identified pathogens. And that's striking. And this is what makes it so scary for how do we even think about approaching this for preparedness. Which viruses do we need to study? What do we need vaccines against? And so if we think about just the known human viruses, there are more than 250 known human viruses. That doesn't count the viruses that are in animals that we know now can pretty efficiently jump to humans. So back in 2017, Barney Graham and I had been thinking about this because I was working on the Ebola vaccine and he was helping with the clinical development, and just realizing that we need to do better. We can't take so long on one pathogen to develop a vaccine. And so we developed the prototype pathogen approach. And that means you've got these buckets of different vaccines that we know can tune the immune response. Now we need to define the pathogens, which is on the other side, so that when we identify a new viral sequence, we've already got the tools to know which one of those vaccines is going to be the best to approach it. So we need to define transmission, pathogenicity, immunity, resistance, and then, of course, surveillance is a huge part of this. So NIAID, amongst many others, thought about how could we go about characterizing pathogens in order to develop prototypes. And the simplest way to approach that was by grouping them into virus families in the hope that within a virus family, one vaccine approach would be the ticket. That's all that would be needed. I want to give you a couple of examples to show that it's not that simple. Again, going back to the need for basic research to understand how to develop prototype pathogens. So one example I'm going to give you is the filoviruses. If Marburg virus had been my prototype pathogen back in the late 1990s, we wouldn't have a vaccine for Ebola virus, because it's much easier to protect against Marburg virus than it is against Ebola. So the immune mechanism needed for protection is different. It was lucky that we started with Ebola virus. The other side of that is not only are there differences within families, but there are examples of a prototype working across families. And so this is on the left-hand side, the structure of the RSV fusion protein. On the far left is the post-fusion protein. So I talked to you about inactivated vaccines, inactivated viruses. Back in the 1960s, RSV was inactivated with formaldehyde. That vaccine was given broadly, mostly to children who are susceptible to RSV, and it actually led to enhanced disease with some deaths. That's because the RSV post-fusion protein makes the immune response make antibodies against the tip of that protein that I'm showing here in the blue, green, and purple. Those antibodies don't neutralize the virus, and in fact, they enhance infection. Work from Barney Graham over many years, again, for RSV, showed that the form one needs to develop protective antibodies is the pre-fusion form of RSV. And so that was groundbreaking for being able to develop a vaccine for RSV. More importantly, it served as the basis for the vaccine against SARS-CoV-2. So the reason we were able to move so quickly to develop an mRNA vaccine is because Barney knew how to stabilize the pre-fusion form of SARS-CoV-2. And so that went into all the vaccines worldwide to give us a highly efficient vaccine. So now this is an example where the prototype works across families between paramyxoviruses and coronaviruses. So what we need, in addition to having those buckets of vaccines, is now to understand at a very basic biological level core functions of viruses that are going to define what is effective for a vaccine or a therapeutic against them. In the case of the RSV and coronavirus, those are both class 1 fusion proteins. So you might be able to put them in the buckets depending on what fusion protein they use. There are non-enveloped viruses that don't have fusion proteins. So there you need to go again to the core functions. What's the mechanism of immunologic protection? How are they transmitted? Where are they pathogenic? Where do they replicate? So all of these things that are absolutely necessary to be able to define prototype pathogens. I'm going to switch for a moment to therapeutics, because I think this is where basic science, again, is equally important for us to be able to develop prototypes for these pathogens. So Ebola virus is an interesting virus. It's evolved many ways to evade the human humoral response. And typically we think about the virus glycoprotein receptor interaction as an efficient way to block virus entry into cells. Ebola is interesting because its receptor is not on the cell surface. So it attaches to cells basically through sticky interactions, and it's rapidly taken up into the cell, as I mentioned earlier. It goes into endosomes, and it's in late endosomes that the glycoprotein gets cleaved, exposing the receptor binding domain, and then the glycoprotein binds to its receptor late in the cell. So what you have is two forms of the glycoprotein, and if you want an antibody to block receptor binding, it has to bind to the full-length form of the glycoprotein on the cell surface and also bind to the cleaved form deep inside the cell to block receptor binding. So that is fundamental basic virology that was needed when I was approaching developing an antibody to block this virus. The next part that I want to talk about and emphasize here is this need for international collaboration, especially North-South collaboration. So J.J. Mayembe is an incredible man who leads the Scientific Infectious Disease Center in the Congo. He has responded to every Ebola outbreak since the first one in 1976. He's a co-discoverer of Ebola. And in 1995, there was a very large outbreak in Kikwit in Congo. And Professor Mayembe had been watching people die from this infection. It had about a 100% mortality rate, give or take. And so he thought, there must be something in the blood of the few people who survive that enables them to survive. He did a very unconventional clinical study where he took the blood from multiple survivors, or the multiple ones that there were, pooled it, and gave it to patients who were infected. It turned out that 7 of the 8 patients who he infused lived. So he basically flipped the mortality ratio. Of course, it wasn't placebo-controlled, and it got a lot of criticism because he didn't do this as a standard clinical trial. But he was convinced, rightly, that there was something in the blood that was protecting patients. So he and I teamed up, and we recruited one of the donors from his 1995 study. Now, this was in the 2000s, many years after this patient had been infected. We took the B cells from this survivor, and then someone who was a trainee of Professor Mayembe came to work in my laboratory. And he used that hypothesis of needing antibodies that bind to both the native glycoprotein and the cleave protein to find an antibody that had those properties. He did that. He cloned the antibody. We tested it in monkeys, and it completely protected monkeys. So then Olivier Babaya, who's another trainee in my laboratory from the Congo, and Subway, when there was an outbreak in 2018, made infusion kits in the clinical center at NIH, flew to Congo, went on motorbikes to the eastern part of the country, which is very dangerous, and infused the first 12 patients with the clinical material that had been through a phase 1 in the US. It was this collaboration that allowed the first treatment for Ebola in Congo. And so that international collaboration is absolutely critical. And it was a public-private partnership with a biotech company called Ridgeback Biotherapeutics. So this notion of needing to collaborate broadly across institutions and widely across nations is absolutely critical for our being able to prevent the next pandemic. So this just shows you the trajectory of the work that was done in close collaboration with our partners. So I just want to leave you with the idea that in order for us to do this basic research that's so important, there are several areas where we need to focus, and I think this is where the efforts in Japan to build these scientific organizations and institutions of training are very important, and that is our ability to define virus transmission, replication, immunologic memory, and I didn't talk about it at all, but I do believe that the chronic sequelae and the host are equally important. So I'll stop there and thank you for your kind attention and turn this over to the next panelist. Thank you very much.
Thank you very much for your impressive talk. Next is Dr. Tetsuo Matano. Dr. Matano, please.
Can you hear me? Okay. I'm Tetsuo Matano, National Institute of Infectious Diseases. And first of all, I'm very grateful to the organizer, in particular for giving me this opportunity to introduce our new organization starting from next month. So the COVID-19 pandemic prompted a assessment of governmental frameworks worldwide, leading to a significant organization reforms to improve crisis responses, preparedness, and capability for future pandemics. According to the government initiative, a new organization, Japan Institute for Health Security, JIS, will be established through the merger of our institute, NIID, and the National Center for Global Health Medicine next month. And so, first, today, I'd like to introduce our NIID and also then introduce new organization, JIS. And our NIID is belonging to Ministry of Health and Labor and Welfare and has 3 campuses in Tokyo, including Murayama campus that has BSL4 facility. And this slide shows a brief history of NIID, and its origin is an Institute for Infectious Diseases established in 1892. This institute was founded by Dr. Shibazaburo Kitadato, and after that, in 1947, half of the function was separated to be our new institute as a National Institute of Health. And the remaining function was, remains at the Tokyo University. So, and in 1960s, it was established to be an Institute of Medical Science, University of Tokyo. And our institute was renamed to National Institute of Infectious Diseases in 1997. And NIID mission is to develop scientific evidence for the control and prevention of infectious diseases in Japan and the world towards infectious disease control. And this institute has 3 missions. So surveillance of infectious disease and pathogen, research and development, and quality control of biological products, including vaccines. The NIID organization was modified after COVID-19 pandemic and now consists of 27 centers and departments. We have 3 administration departments and 3 research centers for public health and 14 centers or departments for pathogen research and 7 cross-sectional centers or departments. Okay. And center or department for pathogen research constructed a basis for the research, and GIS and GIS and centers and departments for host research exert basic and applied research and contribute to GIS 3 function. It means the public health research, R&D, and quality management by collaborating with the centers or department for public health research and drug vaccine development and quality management, respectively. And this slide shows our national network for intelligence on infectious diseases with local research institute, local health center, quarantine station, and hospital. And during the COVID-19 pandemic, the importance of this network was really appreciated. And after the COVID-19 pandemic, GIS network, this network was strengthened, and in the GIS new organization, we need to strengthen this network. And then I'd like to talk about the establishment of GIS. And in September 2023, the Cabinet Agency for Infectious Disease Crisis Management, CAICM, was established as a control tower of infectious disease crisis management under Prime Minister. And also, Department of Infectious Disease Prevention and Control was established in Ministry of Health, Labor and Welfare. And then it was decided to establish GIS, and the mission of GIS is to provide scientific knowledge and evidence for the control of crisis. And government updated the National Action Plan for novel influenza and maybe pandemics, and 13 items were determined, and also the 5 cross-cutting points are also determined. And GIS is expected to have a role, particularly in the number 2, number 3, number 4 for public health, and number 7 and number 9 and number 10 for R&D, and also the number 8 for medical care among these items. And this is the most important slide for my presentation today. So, as I mentioned, NIID and NCGM will be combined to be a GIS, and GIS is expected to be a world-class integrated science center for infectious diseases. And GIS is expected to have 4 pillar functions. And one is disease intelligence. Second is research, development, and innovation. And the third one is comprehensive medical care. And fourth one is human resource development and international cooperation. And for intelligence, to collect, analyze, and evaluate information, it is expected to strengthen emergency preparedness and responses, surveillance, epidemiological research, and laboratory network. And R&D includes basic research, animal model, drug and vaccine development, non-clinical and clinical, TR and reverse TR, and biobank, and possibly domestic and international collaborations. And in medical care, strengthening of clinical network is expected. And for these 3 functions, human resource development and domestic and international collaboration will be a key issue. And I would like to introduce our global activity. And this slide is one of our global activities for human resource development based on the JICA program. And this is JICA NIID Knowledge Co-Creation Program. And this 6-week course has been annually held since 1993 for the laboratory techniques and surveillance. And in total, more than 300 people from the African, Asian, and South American countries participate And we are now constructing network among previous participants by follow-up. Another attempt is construction of Asian laboratory network. This is the AMED project ongoing by our institute, NIID. And it is a project of collaborative study and networking with the Research Institute of Infectious Diseases that play a central role in diagnostic testing, monitoring trends in Asian countries. And of course, this project is expected to contribute to emergency preparedness and responses and strengthening of intelligence on infectious diseases. And furthermore, NCGM is constructing ARISE, ARO network for ASEAN and East Asia. So NCG and also NCGM and NIID are leading a project, JGRID Plus, to connect activity of 11 JGRID centers, which collaborate with universities and hospitals for establishment of clinical research centers. So this will further strengthen this ASEAN network for infectious disease intelligence and R&D, and possibly communicating global network. So in addition, we are making communication with WHO, EC, US CDC, and so on for strengthening intelligence, and with varieties of institute and network for strengthening R&D, possibly towards international clinical trials. And we are, again, we are also contributing to JICA Technique Cooperation Program, and currently in Ghana, Kenya, DR Congo, and so on for capacity strengthening and then R&D. Sorry. And we are also working on human resource development for constructing translational team. Maybe as you know, translational study needs a variety of specialists, including project management, biology and immunology, process development, quality assurance, and so on. So we are now making communication with international foreign countries, and also with the IABI to have a discussion for the human resource development for these varieties of specialists. And now, I'd like to briefly introduce several projects in our institute for the development of vaccine and drugs, possibly contribute to the control of future pandemics. This slide shows current progress on vaccine development led by Dr. Takahashi, Director of Drug and Vaccine Research Center in NIID. In the left panel, an immunogen targeted by memory helper T cells pre-existing in majority of people was determined. And in this SCAD project, messenger RNA vaccines aided with this immunogen are expected to prompt and sustainable antibody responses with enhanced helper T cell responses. In the right panel, they developed a post-fusion HA antigen to induce broader cross-reactive antibody responses, whose clinical trial phase 1 is ongoing in collaboration with Sumitomo Pharma. This group also determined host factors associated with vaccine adverse effects to develop messenger RNA vaccines with less adverse effects. And this slide shows current progress on drug development led by Dr. Watashi groups. They established infection system for screening varieties of drug candidates against individual pathogens. And let me skip explanation. And wet and in silico structural analyses are performed for drug optimization by these groups.
Would you conclude your presentation?
Okay. And in addition, our team led a SCADA project in collaboration with Kirin Holdings, attempting to develop a vaccine to confer innate-based protection against respiratory virus infection. And this is the final slide. So for pandemic preparedness, our institute should have specialists on wide varieties of pathogens. And there are varieties of pathogens with pandemic potential. People recognize the pandemic potential of airborne and vector-borne pathogens. But finally, I'd like to stress the pandemic potential of pathogens that can transmit from asymptomatic carriers such as HIV, hepatitis, and possibly SARS-CoV-2. And it may be difficult to recognize the threat of these pathogens, possibly inducing silent pandemic, but control of these pathogens is an important issue. And in this morning, HTLV infection was mentioned by Dr. Miyazono's presentation, and the silent HTLV spread is now a critical issue. And we are now starting an AMED Aspire program in collaboration with Melbourne University to construct a global consortium led by Australia, Japan, towards the control of HTLV infection. Thank you very much for your attention.
Thank you very much for the important information. The third speaker is Dr. Richard Hatchett. Dr. Hatchett, please.
Thank you. Let me start, Mishima Sensei, to thank you and AMED for the honor of the invitation to participate in the panel today, and to extend my congratulations, CEPI's congratulations, for a tremendous record of accomplishment in the first decade, and many more successes to you. If I could just, at risk of oversimplifying, characterize Nancy's presentation, Matana-san's presentation, looked at the conceptual or scientific basis of response. I think, Matana-san, you spent quite a bit of time talking about institutional arrangements and building strong institutions in Japan. My presentation will really focus on some of the concrete aspects of response. And I think the message that I would like to convey to all of you, I think Jeremy conveyed a similar message in his talk. You need strong science, you need strong institutions, and you need strong execution if we're going to respond to future infectious disease threats. Let me go to my first slide. This is a picture of Sir Jeremy and I in a press conference at Davos in 2020. We're sitting on stage with Ms. Chan, who is the moderator, but importantly, Stefan Bancel, who is the CEO of Moderna. And we were announcing CEPI's first vaccine development contracts focused against the new SARS-CoV-2 virus. The sequences for that virus and the recognition that it was a novel coronavirus had been released only 12 days before. Moderna had been working very closely with the Vaccine Research Center at NIAID, using the prototype pathogen model that Nancy talked about. They had taken the work that they had done on RSV, applied it, as Nancy said, to coronaviruses. They were working at the time on a MERS coronavirus, and it had worked on modifications to the MERS spike protein which they very rapidly adapted to the new sequences of SARS-CoV-2. In fact, when we were announcing this contract, this first provision of financial support to Moderna, what we were funding was the production of clinical trial material. This was 12 days after the sequences were released. The first generation Moderna vaccine was effectively designed in 36 hours from the release of the sequences, showing it was a real proof of concept for the prototype pathogen approach. And I'll come back to that. But it was because of that investment in preparedness and understanding the needs of vaccine development and developing a blueprint for how to develop a coronavirus vaccine that they were able to move so quickly. The vaccine that Moderna produced moved into clinical trials just 66 days after the release of the sequences, but it took almost a year for that vaccine to go through a sequence of phase 1, then phase 2, then phase 3 clinical trials and receive emergency authorization. By the time the Moderna and Pfizer-BioNTech vaccines were actually authorized for emergency use in December of 2020, there had been 68 million documented cases of COVID. That was simply, it was amazing. It was the fastest that a vaccine had ever been developed, and it was far too slow. Subsequently, CEPI became an advocate of something that we called the 100 Days Mission. We thought that with appropriate investments in preparedness, it would be possible to develop vaccines much more rapidly against emerging pathogens to the point that they could be authorized if the risk-benefit context were appropriate at around 100 days. If we had been prepared to execute the 100 Days mission in 2020, 100 days from the release of the viral sequences would have been in April, when there were 2 million cases. You can imagine the number of lives that we could have saved, the economic damage that we could have prevented if we had been able to begin rolling out a vaccine that much sooner. To be prepared for the 100 Days mission, though, we did go back after the pandemic. We looked at all of the innovations in vaccine development, design, clinical trials, execution of clinical trials, innovations in the regulatory approach, innovations in manufacturing. Across all of the vaccines that were licensed in the first 18 months of the pandemic, to see if there were a way to compress vaccine development times using traditional paradigms of vaccine development, effectively using a phase 1, phase 2, phase 3 collection of efficacy data, and then seeking emergency authorization, which is what we did in 2020. For understandable reasons, there were no licensed coronavirus vaccines. Many of the platforms that were being used were being presented for regulatory review for the first time. And we decided that actually, if we took the traditional approach, we could not compress vaccine development much more than was actually achieved in 2020. Perhaps we could go from 11 months to 8 months. But we saw a potential for a shift in how we approach vaccine preparedness that would allow us to reach that target. And it built on the concepts that Nancy and Barney had promulgated starting in 2017 around focusing on prototype pathogens and using that as a foundation within which you would then need to execute a flawless, rapid clinical response and testing approach. The problem, of course, and this slide very much echoes the slide that Nancy showed, is the diversity of threats that we face and the context of increasing rates of spillover from pathogens in animals due to a wide variety of factors, not limited to, but including climate change, population growth, concentration of populations and city incursions into areas that had previously been remote. And to be prepared for future pandemics, we have to be prepared for more than just coronaviruses and flu. We know that. That's where the prototype pathogen approach that Nancy described comes in handy. She mentioned the RSV example. We like to talk about the paramyxoviruses as an example of a family that presents a high level of concern. I think the reason that we like to talk about paramyxoviruses, it's a phylogenetically very diverse family. It contains super infectious viruses like measles and mumps. It also contains super lethal viruses like the henipaviruses, Nipah and Hendra. The concern, of course, would be that there is some virus out there in the paramyxovirus family that would bring both of those attributes together, much as SARS-CoV-2 brought together heightened lethality with the infectiousness of the known cold coronaviruses and combine those in a very lethal and dangerous way. By taking the prototype pathogen approach, effectively, you would be seeking to develop a vaccine design or set of designs, to Nancy's point, that would allow us, and these, what you can see here are clustered, the different genera within the paramyxovirus family. I've circled in the morbillivirus, that's measles, the henipaviruses are Nipah, Hendra, the respiroviruses, the parainfluenza viruses, the rubella viruses, mumps. You could imagine designing a set of paramyxovirus prototype vaccine designs that would cover that phylogenetic space in a way that would allow us to respond rapidly if a new lethal communicable pathogen were to emerge. That's the core concept on the preparedness side. Doing the basic science to understand the designs that we would need to make, doing the development of those prototype vaccines, and testing those prototype vaccines to gain valuable information for regulators will take years. It is a large project across the 25 or 30 virus families that we know can cause disease in humans. It is a large but finite problem, and it is actually tackling that problem is one of the reasons that international collaboration is so important, and I will come back to that. All of that work ideally needs to be done before the next pandemic pathogen emerges, but we should view it as urgent work. It will take us years to do that, but we can do that, and I think we know that we can do it. The second element that we need to bring into our thinking about preparedness, besides this substantial investment in preparedness and in basic science and in understanding what will be required to create effective vaccines, is a focus on execution. An ability to stand up rapid clinical trials, execute them flawlessly, and gather data for regulators within that 100-day limit. We have often talked about the Formula One analogy. It's something that many audiences can relate to. But the idea is that effectively, if you look at pit stops in Formula One, if any of you have watched Formula One races in the last decade, you know that pit stops can be executed in 2 to 3 to 4 seconds. And of course, safety is paramount, even in Formula One. But the way that actually is an evolved capability. If you look at Formula One races from the 1950s and 1960s, those pit stops lasted 60 to 90 seconds. And what happened over time is that the edge that a fast pit stop provided led to an intensive investment in developing the technology, protocols, processes, and skills to actually execute the pit stops, at a much faster rate. They had to redesign the cars. They had to redesign the instruments and engineer the instruments of the pit crew. They enlarged the pit crews. They specialized the pit crews. They developed processes and protocols and practiced those processes and protocols over and over and over again until pit stops could be done flawlessly in 2 to 3 seconds, a 97% reduction in cycle time relative to the pit stops from the 1950s or 1960s. It's actually, I've described this as a deep analogy. The more you look into it, the more you learn from it. And I think it is a reasonable and valid analogy for thinking about what we can accomplish in the clinical trial space if we approach the problem in the right way and seek to engineer the capability that we seek. Now let me conclude just with a couple of minutes talking about a recent experience that we've had in Rwanda. We have been talking about and thinking about and the 100 Days mission since 2021. Our approach to the 100 Days mission was to break the problem of vaccine design, development, and testing down into a series of discrete problems that could then be solved, component by component. We realized that our talking conceptually about the 100-day mission would only go so far. We actually had to get to the point where we were practicing. And in Rwanda, we made an investment with BioNTech, the mRNA company, in their facility that they were building in Kigali. They were building the first full-scale commercial mRNA vaccine manufacturing facility in Africa, and we wanted to extend the capabilities of that facility. Through our engagement with BioNTech and with the government of Rwanda, we also became aware of other capabilities that Rwanda had invested in their own institutions, in the Rwanda Biomedical Center and in the Rwanda FDA, which went from not existing to achieving WHO maturity level 3 in less than 6 years, which is amazing and represents an impressive maturity for a very young organization. But they had also identified critical private sector partners. Ginkgo Bioworks was performing wastewater surveillance on all incoming flights into Kigali. IQVIA, which is a global clinical research organization and data analytics company, was working closely with the Rwanda Biomedical Center. As it happened, IQVIA and BioNTech were already strategic partners of CEPI. So we saw an opportunity to actually work with the government of Rwanda using the 100 Days mission as a paradigm and to think about, could you perform the 100 Days mission in a country? What we envisioned was that we would have an initial tabletop exercise where we brought the public institutions, the Ministry of Health, the Office of the President, and their private sector partners together to begin working over time to think about developing a capability. We imagined that we would engage in functional exercises, actually testing pieces of that execution problem through exercises, with an idea that in 4 or 5 or 6 years, we might actually do a full-scale live-fire end-to-end test against the clock, identifying perhaps a new coronavirus through the wastewater surveillance, creating a vaccine in the Kigali facility, and then testing it against the clock through the Rwanda Biomedical Center. What happened, in fact, was that we had worked with them for about 8 months when we were going to go into our first tabletop exercise with all of the partners. By the time that tabletop exercise actually was scheduled to take place in September, the Mpox emergency in Central Africa, the public health emergency of international concern, had manifested. And so the Minister of Health asked, instead of doing a 2-day tabletop exercise, could we compress the tabletop exercise to 1 day and spend a day on Mpox? And during the day that we spent talking about Mpox, they said, we are about to roll out our first allotment of Mpox vaccine. We are going to roll that out with an electronic medical record system that is new. We would like to do a real-world effectiveness study of the vaccine using our electronic medical record system to perform pharmacovigilance for safety monitoring. Can you rapidly develop a protocol? So what happened was that we rolled immediately into a functional exercise around rapid development of a customized protocol for a specific problem that we got through their regulatory and ethical review and were able to roll out with the vaccine in just 10 days. What happened 4 days after that was a large Marburg virus outbreak in Kigali, a nosocomial amplification of the first cases of Marburg ever detected in Rwanda. And the government called us immediately and said, can we mobilize the partners to organize testing of experimental vaccines against Marburg virus? And we said, of course. And so this is the timeline for the Marburg response. They understood that they had a viral hemorrhagic fever outbreak on the evening of September 26th. They announced that outbreak publicly on the morning of September 27th. We identified a partner, the Sabin Vaccine Institute, that had an experimental Marburg vaccine that was under development by the US government. The vaccine was already in vials in Italy. They had a phase 2 protocol that they were entering into in Kenya and Uganda that they could rapidly adapt. But effectively, we were able to ship the vaccine, adapt the protocol, get it through regulatory and ethical review, train the vaccinators, and perform under good clinical practice, under GCP, a clinical trial that began 9 days after the outbreak was identified. So when you ask, how close are we to achieving the 100 days mission? I think we have proofs of concept on 2 critical elements. We have a proof of concept of the prototype pathogen approach and what that can enable in terms of super accelerated design and production of clinical trial material. And we have a proof of concept that with appropriate investment in preparedness and engagement of partners and taking an approach to actually operationalizing and addressing the logistics of delivering clinical trials at great speed, we can actually execute, even in low-income countries, resource-poor settings in Africa. And so I think if you can imagine a future where we can extend the number of virus families that we are prepared to respond to through investment, and invest in those logistical and operational capabilities, we actually are pretty close to being able to deliver the 100 days mission, which is really, really exciting. But the challenge moving forward, to come back to this theme about the importance of international collaboration, is we have to be able to scale those capabilities across geographies and across the large viral space that needs to be covered. And in both of those instances, that will happen fastest and most effectively if we work through international collaborations. So with that, I'll stop. Thanks.
Thank you very much. We have really impressive talk today. And if you allow me to summarize your talk, is firstly, we need a basic research, continuous basic research. Secondly, international collaboration is important. Thirdly, speed. And to make the speed shorten, we need a kind of a system innovation and process innovation and collaboration across the border. And the fourth one, we have more than 250 human viruses, so we need prototype pathogen approach. Yeah? Is it enough? Anyway, we have 12 minutes, so I need your additional comment after each talk. Dr. Sullivan, do you have any comment? I was really impressed by your braveness. You tried, it's beyond my imagination. For me, it's impossible to study Ebola virus. It's so dangerous and lethal. What makes you so brave person?
So you'd be interested to know that most of the work that I did on that vaccine was at a low biosafety level. So the science has to take place under conditions where you can do robust experimentation and basic research, and that's not in the biosafety level 4 lab. The pivotal studies were done, of course, in a biosafety level 4 lab. But it really is, I think what we need to do is identify the gaps and, again, have a systematic approach to filling those gaps. I thought your Formula One metaphor is just perfect for this situation, and it goes beyond the 100 days mission. It means we need to look at this approach. I think that the fundamental experimental research is an equally intractable problem when you look at it using your standard eyeglasses. You know, we have to be innovative in how we approach that.
One hand, we need a brave person like Dr. Sullivan, and basically research in the site pandemic disease spreading. This is one hand we need. Another hand, we need an organizer, leader like Dr. Hatchett, lead us to the next phase. Dr. Hatchett, would you comment a little more?
Well, thank you. I mean, one comment that I would make just is that I do think even by taking the prototype pathogen approach, we're talking about needing to develop dozens of vaccines against dozens of viral families, and that task is simply too large for any single institution to undertake. But if we work collectively, if we communicate and share information, I actually think we could complete that task probably easily within a decade, maybe faster. And I want to just commend Hamaguchi-san. I just want to commend SCARTA for the collaborative way that you have reached out to other institutions charged with developing medical countermeasures. For the audience members, SCARTA will be hosting a meeting tomorrow and the following day of a informal medical countermeasure funders roundtable that's been gathering now for a couple of years to share information about the individual institutions' priorities, to raise common problems for discussion, and we're really grateful to SCARTA for taking that initiative.
I should say thank you. Because of your support, we can make it possible. So anyway, Matano-san, can I have your comment, additional comment? You think you're on the way to make a system innovation in G's. You think, is it successful? Directly ask.
So actually, so in the new institute, for the R&D, two key issues are expected. And one is to have a basic research system that can respond to a wide variety of pathogens. And this is related with your presentation. And the second one is to establish a system that enables rapid and efficient flow from the basic research to seed development, non-clinical and clinical trial towards clinical use. And that may be related with the presentation. And in our system or in Japan, for both of those, one of the key issues is human resource development. So we need really recognize the importance of human resource development after COVID-19 pandemic. And another comment is related with strengthening the infectious disease intelligence. And because infectious diseases are affected by social activity, so it is important to establish integrated science that includes not only physics and so on, but also the social sciences. And I believe it may also contribute to the progress of strategic communication to the control of infectious diseases. That are my additional comment.
Thank you. Unfortunately, time we can share is limited, and we have to close the discussion soon. So I would like to hear final brief comment from each speakers to conclude this section. What is the most important key word for you to solve the, or I would say, prepare the complete preparedness? Dr. Sullivan, please.
I would be glib and say prepare. That's the key word. But no, I think it is not an intractable problem. I think you heard some ideas this morning or earlier today with Sir Jeremy's presentation and my colleagues here in the panel today, that it is a problem we can solve by putting the resources and the organization and the partnerships in place that will be necessary for us to do this.
Collaboration. Dr. Matano, please.
We still have a lot of mechanisms of something that are not elucidated, and recognize that the key points are, again, the development of varieties of human resources.
That's important. Dr. Hatchett, please.
So just to echo the themes, first, I do think that for most viruses, not all, obviously, we're still working on HIV, 40 years after it was discovered. So not every single virus is going to be tractable to the approach that we've laid out. But I think that for most viruses, this is a solvable problem and is a challenge to humanity in the 21st century. This is a much easier problem to solve, for example, than the problem of climate change. But what it will require is sustained effort and will and attention and commitment to collaboration on multiple levels, not just the scientific level. I would say, looking back on the COVID experience, the one thing that worked very, very well during COVID was the collaboration in the scientific community across borders. That was exemplary. There were a lot of things about the COVID response that were not. I should just say, because I neglected to mention this earlier, I should say that WHO's role in elevating the pathogen family approach in their latest update to their pathogen prioritization process is hugely important, because it sets a normative standard for the world to gather around and to begin this collective work. And I think we have to maintain our support for WHO as an institution. It's going through a very challenging period. But if we're going to solve this problem, which we can solve, we have to work together under these kinds of multilateral efforts and organizations. And I'll just stop there.
Thank you very much. I would like to emphasize again, international collaboration is our start point, essential work. So we have to work together. Tomorrow we will have round table discussions. Let's talk each other about these important issues. Anyway, thank you very much for your valuable information and a nice talk. Lastly, I would like to give the speakers a big round of applause. Thank you very much.
Thank you very much for the presentation and discussion. Thank you very much.